All right, I think we should get started. Hello, everybody, and good morning, good afternoon, good evening to everybody joining in from around the world. Welcome to our first workshop in the international series, How Does Sharing Genetic Sequence Data Impact Biodiversity Science and Conservation? My name is Jocelyn, and I am the Director of Public Policy at the American Institute of Biological Sciences and Executive Director at the Natural Science Collections Alliance. I am also a member of the planning committee that has taken the lead in organizing these workshops. Before we get started, I just want to go over a few logistical items. This workshop is being recorded, and as backup, the webinar is also being live streamed privately on YouTube. The YouTube video is currently private, so it's not searchable or available publicly, and the recording will be posted and shared after the workshop. You can use the chat feature, which is at the bottom of your Zoom screen, to communicate with people on the call, including the panelists and other attendees. You may use the chat to introduce yourselves with your name, affiliation, and the country you are joining from. If you're experiencing technical problems, please reach out to IT support Diane over chat for assistance. After we hear from our speakers today, we'll have a Q&A session to address audience questions. So if you have any questions that you'd like to ask, please type in your question into the Q&A box, which is at the bottom of your Zoom screen. Now, this workshop series is sponsored by the National Science Foundation and led by the American Institute of Biological Sciences in collaboration with the USA Nagoya Protocol Action Group. As you can see here, there are several scientific societies that have partnered with us in this series, and we deeply appreciate all the support we've received from our sister societies. This workshop series is organized around 6 broad topics or themes. And today's workshop is focused on applied ecology and infectious disease. As you can see on the slide, we have an exciting slate of speakers on the program today who will be sharing their experiences and perspectives on navigating international research collaborations in the current policy landscape. Specifically, we'll be hearing about emerging pathogen research across the Americas with speakers from Argentina, Brazil, Mexico, and the United States. I'd like to thank the American Society of Mammalogists, the Natural Science Collections Alliance, and the Society for the Preservation of Natural History Collections, or SPINACH, for co-hosting and participating in today's workshop, and for working with the organizing committee in helping this program come together. A special thanks to Joe Cook, who has been instrumental in developing today's program, and who will also be facilitating the discussion later today, along with John Bates. And I also want to offer my gratitude to my fellow organizing committee members, John Bates, Rachel Mayer, Rebecca Adler, Mr. Andino, Crispin Taylor, and Breda Zimkes, all of whom, or most of whom, are on the call today helping with facilitation. Now, the main intention behind these workshops is basically to increase awareness among biodiversity scientists in the US as well as internationally about ongoing negotiations and international policy discussions around the application of access and benefit sharing principles to digital sequence information. Now, we will learn more about the terms that I just mentioned and about these ongoing discussions in a few minutes. But through these workshops, we also want to provide a platform for the research community to learn from the experiences of others about best practices for collaborating internationally on biodiversity research. So to give you an overview of today's program, after a brief introduction and remarks from our host societies, we will go over some background information about the current policy landscape, and then we will get into speaker presentations, which will be followed by a Q&A and discussion session. I am now going to ask my fellow organizing committee member, Rachel Mayer, to talk about the Nagoya Protocol Learning Portal. And Rachel leads the development of this website and will provide a quick introduction to the community discussion forum that we've set up for this workshop.
Thanks, Jocelyn. Hi, everybody. It's so awesome to see so many people here from around the world. Really excited to have a stimulating workshop today. So learnnagoya.com was a little website that some of us started with the US Nagoya Protocol Action Group. This was a whole bunch of representatives from societies, different scientific societies that came together to try to build some educational resources, because we realized that a lot of our members were not really privy to what the Nagoya Protocol was. And it was hard to find sort of less jargony educational materials that you could, say, use to teach a class or to introduce this to your students who might be just starting to engage in international fieldwork or working with international collections. So we built this website. It's still sort of a work in process, and we have a workshop page that you can find on the homepage. So if you, Jocelyn, if you go to the next slide, there's the middle button. You can see the workshop title to what you're at now. You can click on that, and at the bottom of that workshop page, eventually we'll host the video there. But at the bottom of the workshop page, they have another gray button. It says, add to the workshop discussion forum here. And that will pull up a FramePad or Framapad. This is a really cool open document you don't have to sign into. So it is sort of based on that you're going to be nice and not erase someone else's comments. So everybody you can make up whatever username you want. So it can be totally anonymous. This is a space for us to just jot down our thoughts. We have some charge questions for you. And this is a place where you can also put additional feedback or questions for the speakers. This document will live on for 6 months after this workshop. So we promise to keep bothering hounding the speakers that if you have a question in a couple of weeks that comes up, that we make sure that we get that answered for you. So this is your space. You can also chat with each other. And we're going to be collecting all of these and ideas and really trying to kind of synthesize what this community wants. So, all right, that's it for me. If you have any technical issues with that, we have Mega who is here, and I'm here, and lots of other people are here to try to help you with that. But that's going to be an active space besides Zoom that you're going to be able to use for the day and beyond this workshop as well. Thank you.
Thank you, Rachel. I am now going to yield the virtual floor to our host society reps to say a few words and then introduce our speakers. First, we'll hear from Deanne Reeder at Bucknell University, who is representing the American Society of Mammalogists. And then we will hear from Dirk Newman at the Bavarian Natural History Collections, who is representing spinach. Go ahead, Deanne.
Thank you very much. I'm really pleased to, on the behalf of ASM, to welcome you to this really important workshop. If we can go to the next slide. ASM has been around for just over 100 years, currently has about 2,500 members. Many of those are working in museum communities or doing field research. And the ASM is really engaged in trying to facilitate the needs of committee members, mostly through the Legislation and Regulations Committee. We're a member in the USA Nagoya Protocol Education Action Group, but we also have other committees that interface with Nagoya and with issues surrounding digital data. And so I'll pass that on. I'm really happy again for ASM to be a contributing supporter of this.
Thanks, Deanne. Go ahead, Dirk.
The next slide. So welcome, everybody. My name is Dirk Newman. I'm representing the Society for the Preservation of Natural History Collections. This is actually a group of very active and engaged people from the museum and collection care group. Next slide, please. The society has different committees. I'm co-chair of the Legislation and Regulations Committee. We have other groups, or members in the group are also active in the US Nagoya Protocol Education Group. We have done several submissions to the CBD Secretariat on DSI and conduct specific workshops and trainings, among others, for Nagoya and ABS at Spanish conferences. There are also 2 other committees engaged with Nagoya questions, which is the best practices committee. They have a very nice website which has a lot of valuable information on the Nagoya Protocol. You can see the link or the website address there, and also very active and with the status of an official observer is our Biodiversity Crisis Response Committee, and they follow the current processes closely. And I have the pleasure now to introduce our speakers. So first is Jocelyn Colella, based at the Biodiversity Institute of the University of Kansas in the United States. She co-founded a virtual group of museum that looks into museums and emerging pathogens in the Americas in 2021. And that group aims to connect biorepositories with biomedicine research. She is a curator of mammals at the KU. Gerardo Susan is a veterinarian and based at the National Autonomous University of Mexico. He is a member of the National Mexican Research System, SNI, and has been appointed recently as the representative of Mexico for IPBES, that is the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services. Marcelo Weksler is at the Museu Nacional, Universidade Federal de Rio de Janeiro in Brazil, and part of the Nagoya Working Group. His research focuses on small mammals such as rodents, marsupials, bats, and their role in harboring zoonotic physiological agents. And we start the presentations with Luciana Silvestri. She is an lawyer and specialized in environmental law research at the National Scientific Technical Research Council, CONICET, in Argentina, and she has served as legal and technical advisor to the Spanish Ministry of Environment on different Nagoya and ABS questions, but also internationally. She served as an expert, for example, as an expert to the Informal Advisory Committee of the ABS Clearinghouse. And she will give us, hopefully, a good start in this workshop and give us some common ground and understanding on digital sequence information, the Nagoya Protocol, and what's it all about.
Thank you. Well, good morning, good afternoon, good evening, everyone. It is my pleasure to share with you this presentation and to have the opportunity to engage in debate with all of you. I will get straight to the point, and therefore I will briefly outline the basics for understanding DSI, also known as digital sequence information. Next slide, please. Until recently, research and development on genetic resources necessarily implied access to genetic material that conveyed or supported the information of interest. For example, it was necessary to have a set of cells to extract genetic information from them. It was also necessary in certain cases to collect, hunt, or remove the organism, whatever it was the case, to get access to the genetic resources in question. Although today access to genetic material and collection of biological resources remain fully valid practices, researchers increasingly rely more and more on the use of digital sequence information to achieve desired results. The phenomenon of DSI would comprise, in principle, information related to millions and millions of genetic sequences, which is stored in a digital format. The term DSI is a placeholder term, which is mostly used within the framework of the Convention on Biological Diversity. Still, there is no consensus about the term. In other areas, especially within the scientific community, the preferred term is genetic sequence data. Most importantly, what is or should be included under the term DSI greatly varies. For some parties to the convention, DSI should not only include DNA and RNA sequences, but also amino acid sequences and metadata associated with them. For other countries, DSI should only comprise DNA and RNA sequences. In any case, what is important to note is that DSI is the result of a physical access to genetic material and of a subsequent reading of the sequences. Once information is digitalized and stored, researchers can access it at any time from any place, and in many cases, free of charge. Next one, please. DSI is under discussion in the framework of several multilateral instruments. A few of these are the ones you can read on the slide. All of these forums and organizations focus on different competing priorities and interests that must be balanced. For instance, conservation of biodiversity, sharing of benefits arising out of the utilization of genetic resources, intellectual property, equity, technology transfer, etc. We will focus on the debate that takes place in the framework of the Convention on Biological Diversity and the Nagoya Protocol, because these two instruments are the cornerstone of the entire legal regime on access and benefit sharing and biodiversity conservation. Next one, please. The fundamentals of the CBD and the Nagoya Protocol in regards to access to genetic resources and benefit sharing, a regime known as ABS, are: first, in order to access to genetic resources, you must get permission from the provider country, known as prior informed consent. Second, you will need to negotiate an agreement to share benefits resulting from the use of genetic resources. Finally, all parties to the Nagoya Protocol shall take appropriate measures to ensure that genetic resources utilized within their jurisdictions have been accessed in accordance to PIC and MAT. Next one, please. Despite the benefits that the utilization of DSI entail for research and development, its use is controversial under the framework of the CBD and the Nagoya Protocol. In particular, parties debate on two main issues. First, whether DSI is included or not in the scope of both agreements, or at least within the scope of the Nagoya Protocol. Second, parties disagree on the implications a possible inclusion or exclusion of DSI could entail for the fulfillment of the agreement's objectives. The problem is not minor. If DSI is understood to fall out of the scope of the Convention and the Nagoya Protocol, provider parties would not have the right to benefit from own genetic resources once the information they contain has been digitalized and stored. On the contrary, if DSI is understood to be included within the scope of the Convention and or the NP, a series of complexities would arise for the implementation of the ABS regime. These complexities include difficulties to determine the origin of the genetic resources from which DSI was originated, complexities to monitor the use of DSI, and problems to determine the extent to which each sequence has contributed to the final result for which benefit sharing should be triggered. Next one, please. Countries' position regarding DSI can be summarized into two antagonist views. On the one hand, the view held by Brazil, Ecuador, Mexico, India, South Africa, Argentina, Ethiopia, and other countries which are mainly considered providers of genetic resources. The second group of countries includes Canada, the European Union, Japan, Australia, and Korea, which are mainly regarded as users of genetic resources. The fundamental point of view of provider countries is that DSI is already included within the scope of the CDB and/or the NP. Therefore, benefits generated from its utilization should be distributed between the user of the information and the country in question. To support their point, they assert that genetic resources were, are, and will be valuable precisely because of the information they contain. Their point lies in two premises. First, the notion of justice. Second, the concept of sovereignty. Justice calls for benefits from DSI to be distributed, since DSI was and could only be generated in the first place thanks to genetic resources found in their territories. As for sovereignty, it would be a contradiction to exercise sovereignty over genetic resources as they do and have the right to participate in the benefits generated from their use, but not to have the right to participate in the benefits that arise from the utilization of DSI. The central issue on the agenda of user countries is the need to agree on open access and free circulation of DSI. They believe regulations should not be imposed because they would hinder access and use of DSI. Two main premises lie under their discourse: the idea of scientific progress and of freedom. In their reasoning, freedom leads to scientific progress. In addition, the idea of freedom, as opposed to control, is linked to the principles of and efficiency. In their view, the nature of the good to be controlled, that is information, it makes it impossible to control and ineffective and inefficient. Next one, please. Discussions on ABS and DSI are ongoing in the framework of the CBD and the Nagoya Protocol since 2016. Decisions of the parties establish a science and policy-based process. The process entails the submission of views and information from parties, other governments, indigenous and local communities, actors, and relevant organizations, the commissioning and peer review of 4 different studies, and the establishment of an ad hoc technical expert group on DSI. Recommendations by the working group on how to address DSI in the context of the post-2020 global biodiversity framework were considered by the Conference of the Parties at its 15th meeting, which took place a few days ago in China. The meeting will be resumed on April next year. Most likely, at a certain point, there will be regulation on DSI. The question is how it will be covered. Next one, please. So, how can the scientific community engage in the policy debate? Here are some recommendations. First of all, make use of every opportunity to explain your point to negotiators in a clear and comprehensive manner. In addition, as mentioned in the previous slide, relevant organizations can submit their views to the CBD Secretariat whenever there is the opportunity to do so. Second, try to empathically understand the reasons behind provider countries' position and to propose solutions that allocate their interests to. Third, get in contact with your peers from the north and from the south. You are all scientists, and the message, interest, and reasons of the scientific community should be conveyed as one, taking into consideration common ground, but also differentiated capabilities. In so doing, key approaches to the issues could, first of all, emphasize the distinction between commercial and non-commercial use of DSI. Second, propose and develop mechanisms for responsible use of DSI in the framework of non-commercial use of DSI. Finally, find and propose creative benefit sharing opportunities, because benefit sharing is not always about money. Thank you.
Thank you, Luciana. Next up is Marcelo Weksler.
Thank you, everybody, for attending this workshop. Also like to thank the American Society of Mammalogists for inviting me, Diane and Enrique. Thank you also to Joe Cook and John Bates for the invitation. Basically, thank you, Luciana, very much for your presentation. It clearly established what we are dealing here in terms of digital sequence information. Joe and John asked me to give a more on-the-ground approach, detailing a little bit how Nagoya Protocol in general is affecting the basic biodiversity science in Brazil in a personal case, but I would extend this experience to a lot of my colleagues. Just next slide, please. Just to put in a context, our research, or the research that we develop at our lab, we are dealing with basic research on the biodiversity of our country. It's a mega diverse country, Brazil. This is a picture of what's going on in our part of the world. This is Rio de Janeiro, this big urban area, and the remaining is part of the landscape. Very, very small patches of conserved forests. Almost all of those are in national parks. And we still have a vast gap on basic biodiversity knowledge there. In the past 10 years, we described 2 new genera of small mammals, of rodents from this area. And this is one of the most studied areas in Brazil. So we are still discovering new mammals on the best known area in Brazil. Imagine other areas such as Amazon and other parts of the country. This is a very interesting area, a lot of environmental gradients, and we're still doing the basic research on that. Please, next slide. I just came back from the field, from field work on this area. That's one of those national parks there. It's Parque Nacional de Itatiaia. A lot of our work is done in collaboration with folks at Fiocruz. That's the major, I'll say, public health institute in Brazil. And our current project is really looking at basic data for zoonotic agents in small mammals, bats, rodents, and marsupials. We are trying to identify the species. We are trying to see if there is any altitudinal pattern on the distribution of both the mammals and their parasites, so endoparasites, ectoparasites, viruses, bacteria. We are trying to, next slide, please. We are trying to collect as much information as we can from those creatures that we kill, we sacrifice. So we believe firmly on the holistic specimen approach. We try to, actually, some of the folks in Fiocruz, sometimes we are called to outbreaks, to hantavirus outbreaks or arenavirus outbreaks. So it's sometimes we go into situations that it's not optimal to work, and we try to, from that situations, try to extract the best kind of data. And we are describing not only the mammals, but also their parasites and so on with our specialist colleagues at Fiocruz. Next slide, please. The point I want to make here is that all that basic biodiversity research that we do, our central method of research in contemporary taxonomy is integrative systematics, and central to that is it's the sequence data. I like to call it digital sequence data. I'll show that we refer that at in silico information also, but most of our research, or a central part of it, it's based on phylogenetic analysis of molecular data. Several of the taxa that we work with are very hard to differentiate morphologically. We have several cases of cryptic species groups. We did some analysis from papers published in one of the major journals in the area, Zootaxa. This is about 70 new mammalian species described in the last 20 years about, and 70% of them work with molecular data or sequence information in their description of new species. So this is central. And this is not only the digital sequence that the researcher created, but also obviously sequence available freely, publicly in public database, the three major databases in the US, Europe, and Japan. Next slide, please. On the other hand, it's really, really hard to transfer that information from field work to really producing genetic data, or the end product, in our case, is depositing this sequence in GeneBank or so on. The red tape, most of you will know in several countries, is enormous. It's redundant. We have to go and fill several federal forms online through our university system. The ones that I will talk a little bit more here is the one term CISGEN, is the national system for genetic information that we have to fill in several steps of our pipeline for genetic data production. And several of them are redundant with existing regulatory framework. So I'll not complain a lot of this, but just to say that even before Nagoya Protocols kicked in Brazil, and I'll show you a timeline, we already had several regulations in place to perform our research, from collecting, from archiving it in biological collections, doing the proper research sequence. And I will not mention exporting. I'll mention later because it's probably the worst part. Let's go to the next slide, please. The context here of my talk is also have to be based on my work as a curator at the Museu Nacional in Rio de Janeiro. I'm curator of mammals there. I'm taking care especially of the tissue collection. I also work with the vulture collections, the largest in South America. Very important to mention is about 3/4 of our collections came from collaboration with public health institutes such as Fiocruz. Even before that, since the 1930s, we have been collaborating with several expeditions or work with zoonotic episodes like the yellow fever, malaria, and even bubonic plague. So about, I'll say, 3/4 of our collection come from those collaborative work. We at the museum, I don't think I'll need to stress this in this public, but we are central to the research, outreach, and the archival of the data that is being produced now in the past and that will be used in the future. So it's the central nexus for long-term biodiversity knowledge. Next slide, please. I'm sorry. The vast majority of collection, at least in South America, are underfunded. I'll just go quick on that. We are conducting a survey on our mammal collection in South America, and we have some data that corroborate that. Here in Brazil, our universities do not have resource. Most of them do not have resource for their collections. There is no specific line of funding for collections nationwide. And from CNPq, that's our major financing agency here. Some states do have, but most money to maintain the collections will come from our own research projects. We'll also have some funds to buy alcohol, for instance. It goes as basic as that. Next slide, please. So just to give a quick timeline of the implementation of the CBD treaties and Nagoya protocols here in Brazil. We have very early in this century of the first legislation regarding that stayed in place until 2015. During that time, the community the scientific community got a very good interlocution with Brasilia, with bureaucrats, and we got this resolution 21 that was very important at the time, that exempted the researcher working with taxonomy, ecology from dealing with that kind of bureaucracy. But starting with the new legislation from 2015 to now, we are not any longer exempt from that. We have a lot of red tape now under our backs, and the situation is, I will not say catastrophic, but because we try to deal with that. But since 2015, we have a very shaky regulatory framework, because we have several resolutions changing the rules. We have a very bad, poorly written decree that creates a lot of problems. I'm gonna mention some of them. This year, we finally ratified the Nagoya Protocol. And this year also, after 6 years, I'll put 5 years, that's when the decree was made, we at the University in Rio de Janeiro finally signed our first mutual transfer agreement. So it took us 5 years to come up with a model for exchanging specimens, engaging in international collaboration. So let's go to the next slide, please. Just quickly point some of the major issues here. Regarding DSI, properly speaking, our regulatory framework already consider, as Luciana mentioned, in silico or DSI, digital sequence information, as part of our genetic heritage. Brazil equated genetic resources from CBD with genetic heritage, so it's any kind of information on genetic origin. So even phenotypic data, even if you wanna go to the museum and analyze specimens there, you have to fill out forms for access of genetic data. So also in silico sources, sequence from GenBank or the database should be registered. If you access Brazilian data in GenBank, in theory, you should, first of all, get in contact with a Brazilian researcher, so he or she will be able to put you in their project. If you wanna, registration must be carried prior to the shipment or to the access of the data. You have to fill out an online form, very complicated form. We have been complaining about that form for years now. Maybe we'll get some simplification on that. And finally, the most important in this context here is that access to genetic heritage is prohibited for non-Brazilians. So any researcher that wants to do work with Brazilian material would have to collaborate with somebody in the Brazilian Institute to access that specimen or genetic information. You can have more information on those links here. And that's been creating several problems. We can discuss some of them. It has creating problems with the International Committee of Systematic Parasitology. There are rules on type culture, request that new species should be deposited in 2 accessible culture collections in different countries. So Brazilian researchers are not doing that, so they are in danger of not being able to describe new species for parasites. And several other cases of researchers having to create or employ loopholes in conducting their research, trying to get specimens out of the countries. We know people are getting fined because of that, and we hope that the situation will get better, but right now we are in a kind of limbo in terms of this regulatory framework. Next slide, please. I think it's the last one. I'm sorry if I talk a lot. We have been complaining, or we'll say the community in Brazil of researchers doing basic research in biodiversity, they occasionally get together, we write papers about that. This one here from South Africa, it's the same from our colleagues, just came out in South Africa, explaining clearly that the red tape it's harming and threatening, strangling our research, our basic biodiversity research. And this actually helps. So we are able to get in contact with our representatives in Brasilia, the academic representatives in these negotiation rounds. Just 2 days ago, a new decree was released, a lot of it based on our suggestions. We'll see how that will work out, but we will have new regulations for basic non-commercial scientific research. Next slide, please. No, excuse me, sorry. Yes, so just to send up the current regulation in Brazil, it's not working for basic biodiversity science. It imposes a lot of bureaucratic red tapes that in the end we'll have to deal with because our institutions, except a few of them, do not have a good bureaucratic personnel to deal with that. So in the end, researcher and students end up with more bureaucracy, and we don't have the benefits. My opinion, that's personal, and several colleagues agree, it should remain as open and freely accessible as possible without commercial users to be explicit whenever data is deposited or used. And finally, biological collections provide this structure for long-term preserving genetic resources and should be central in this benefit sharing distribution. Thank you. We can discuss this later. I'll pass the presentation to Gerardo Suzanne.
Thank you, Marcelo. Well, I will continue with some topics similar like Marcelo presented. Next slide, please. Well, this is a very timely meeting because we are right now in a pandemic, and this pandemic has been recognized that is by different institutions as a result of these changes we have done in the world. And we need to develop strategic ways to identify genetic diversity of pathogens. We need to develop international collaborations and to develop a justice in sampling, justice for local people, for local communities to get information, and capacity building for several countries in order to get important information for biodiversity and important information for infectious diseases and important information for modeling infectious diseases. So this pandemic, next slide, please, has been the result of different interactions that we are doing in the world. And this information that we can get from genetic diversity of pathogens and hosts, and if we can start to work together. Next slide, please. To understand how these changes occur, it has been several studies have identified that these anthropogenic changes are occurring worldwide, and several ecological aspects as infectious diseases emergence is occurring worldwide right now. So we have to track this in a proper way. Next slide. So this, for example, this is an example as a nice collaboration we have between US and Mexico, that even though all the hard procedures to export samples to US and to develop some metagenomic studies, we can identify several family of viruses in different places in Mexico, in central and southern Mexico. And this allows us to develop models, to develop a nice collaborations. And this is an example of what we can do if we get easier all the procedures and information. Next slide, please. So what we have, for example, in the relation with this, all the habitat destruction, habitat modification, with this collaboration, we can identify, next slide, how the viruses are assembled in different places, and we can identify how different coronaviruses have been getting together in some places, and we are changing. So these are examples of this that we can do together, identifying how and when and what kind of viruses can emerge in some places or not. So this could be a nice stuff to continue this. Next slide, please. So what we have done, because there are a lot of limitations between exportation and importation and sharing data, is obtaining data available and to develop models and to develop how these species occur, how these infections occur, how vectors occur, and identify risky areas. But if we have really the genetic information, if we can share it, we can develop more precise models, and we can develop a better opportunities for doing research and identify risky areas. Next slide. So what we have done in the past is a very hard work. In all procedures we have to export. We have collaborated with different countries in Central, in South America, in North America, in Europe. And the problem has been a long way to export a sample, to get information. So it has been really a hard work. And there are a lot of permits for local permits, international permits, as Marcelo mentioned. Next slide. So we have dealt with that, we learn of that, and we have different critical points with the samples. We didn't follow the cold change, the costs were very high, delays, the tension in customs. Sometimes the samples stuck there for months, and we lost all the hard work. So the Nagoya Protocol, next slide, please, represent an opportunity for several aspects, no? Represent an opportunity to identify pathogens, to model different risk scenarios with international and intersectorial collaboration. The problem is the intersectorial collaboration, no? That we need to enhance intersectorial collaborations. We are enhancing in the academic way the international collaborations, but when we get stuck in the national health, animal health system or the human health system, there are not nice collaborations, and sometimes it's difficult to work when you don't have an intersectorial collaboration. But this protocol will allow us to develop more information, to avoid the biopiracy, because we have in Mexico a lot of species, as mentioned before. Several species have been stolen, but also in samples, even also in pathogens. Sometimes other countries get the information and publish data, and we don't have the information here. So we have to develop capacity building. We have to develop more justice in these biological samples to work on this. No? So we have several examples. And in Mexico, we have another limitations, that we need to implement administrative and legislative policy measures that allow its operational and in a national territory. We have to work on that. We have to work academy, institutions, museums, the government to work on this and to do a better job on this. And we don't have enough legislation on these topics. Next slide. So what we want to work is in a project. This is an example of a project we are working right now here in the Peninsula of Yucatan in Mexico, is to have a different multidisciplinary approach. And it could be an example of what we can do if we continue. Next slide, please. This project, we want to include how the systems are collapsing, how the systems, the viruses are assemblishing, in which species, but doing the same in different countries and having this international collaboration will allow us to have a better prediction of macroecological problems, macroepidemiological problems. We can do several things that could be a very good help for the infectious diseases prevention and infectious diseases monitoring. So we are trying to develop this web of communication between countries. And obviously, we don't have solved all the sharing genetic material. Next slide, which could be a nice situation. But also we want to work together under different approaches, having local people, having social scientists, having epidemiologists, having modelers, having biologists working on the information in order to get the better information for all this genetic material and have a better tool to predict and prevent and to work with people and include people on their principles of justice and other aspects, respecting the land and other aspects, the culture. Next slide, please. So having approaches, ecological, social approaches, evolutionary approaches, biogeographical approaches to understand all these processes will be better if we have all the protocol, the Nagoya Protocol as is established, and we hope we can get it in near future. All the facilities to do a best sharing genetic material. Next slide, please. So we need new approaches. It's not only to sharing data, it's how we are going to work with this data, how we're going to work in a justice, in an equal way to collaborate, respecting the land, respecting different places. And this is an idea, how can really help in an international, in a local way, in the national way. So we need to have different approaches to work on this and think on this also, as an important part of our research. Next slide, please. And thank you all the collaborations, people who have been an important collaborations with different countries, with different institutions. And this is a nice example of how, if we do all the proper sharing genetic material, will help a lot for the infectious diseases in general. So, well, the next speaker is Dr. Jocelyn Colella. Thank you, and see you.
Thank you. We can just jump right into it. We'll go to the next slide. So I'm Jocelyn Colella, and that was a wonderful introduction by Luciana, Marcelo, and Gerardo. Gerardo and Marcelo both identified a number of challenges that they've encountered navigating Nagoya in their countries. But as the last speaker today, I want to address a potential solution. I'm in the United States, as you can see from this map, and the US is the only country that has not yet ratified the Convention on Biological Diversity, or CBD. It might seem strange then that I'm here from the US talking about Nagoya, but because every other country in the world is involved, if US scientists are doing any international work, it's because they have to navigate Nagoya also. So we are dealing with this too. Next slide. To be honest, the US is part of the reason that legislation on benefits sharing needs to exist in the first place. There is a long and ugly history of colonialism and political and economic exploitation that extends into the products of science, and we can't ignore that. Disproportionately, wealthy northern countries have traveled to, extracted, and exploited the resources of lower income countries, predominantly in the global south. And Nagoya aims to prevent the continued exploitation of these countries, which is a good thing. Next slide. Nagoya was founded in response to commercial exploitation, though, which is a very specific subset of DSI and genetic material users. For instance, briefly, in the early 2000s, Indonesia asked the US for help responding to an outbreak of H1N1. Without permission, the United States Centers for Disease Control, or CDC, passed samples received from Nagoya to big pharma drug manufacturers. Big pharma made a vaccine and then tried to sell that vaccine back to Indonesia for a profit. This is clearly exploitation. Next slide. In response, Nagoya legislation has focused mostly on provider countries receiving financial benefits from users. And while this might make sense in a commercial framework, it is incompatible with non-commercial research in multiple ways. Next slide. Thank you. First, what is a provider country? The colonial definition, which is unfortunately often used in Nagoya, assumes that some countries are providers and others are users, but this is not the case. Let's look at the next slide. These pie charts were published by Scholes et al. This year and show the locations of DSI users and DSI providers. And without labels, I think it's pretty difficult to tell which is which. I can't really. If we look at the next slide, we see that users are on the left and providers on the right. Both of these classes are international in nature and often reciprocal. The binary interpretation of mega-diverse developing countries as only providers perpetuates this old model of colonialism and is simply inaccurate. Next slide. This colonial view of provider countries, combined with the limited view of benefits sharing being only financial in nature, treats genetic material and DSI as another commodity, when in reality, we need to be working towards mutual collaboration, integration, and justice, as Gerardo just emphasized. Although governments and companies may not agree with me, there are other benefits besides money. Next slide. For instance, training of in-country personnel, capacity building and research, development of local scientific infrastructure, and usage tracking are just some of the other types of benefits that can be shared. But these types of benefits typically don't come from commercial users. Next slide. So while financial benefits sharing might make sense for commercial users, next slide, I have duplicate slides here. The financial, I see duplicate slides, but I guess there's not. Sharing of financial benefits is simply going to be intractable for universities, museums, non-governmental organizations or NGOs, and other non-commercial research organizations that also use genetic materials and DSI in research and education. We as a scientific community need to emphasize and communicate the difference between commercial and non-commercial research to our representatives so that these interests can be considered accurately in the legal formation of these processes or policies. Next slide. To get ahead of this, we have formed a virtual community of practice called Museums and Emerging Pathogens in the Americas, or MEPA. Because this legislation affects us all, we need a unified and international voice. This message cannot just come from the US. MEPA aims to unite biorepositories across the Americas with ongoing biomedical and pathogen surveys. Next slide. With the goal of acting as a global decentralized pathogen surveillance network by both ensuring preservation of and access to wildlife samples. The issue of access across international borders has made Nagoya and the legal treatment of biorepositories, DSI, and genetic materials a major point of discussion in this group. Next slide. For those of you that do not work at biorepositories, these archives, which can include liquid nitrogen frozen tissue collections of wild animals, represent primary infrastructure for emerging disease research and response. Often, biorepositories have the only available pre-spillover samples, And by linking pathogens and their hosts in space and time, these resources allow scientists across the globe to identify biotic and abiotic factors that contribute to spillover and govern the evolutionary dynamics of pathogens in both wildlife and humans. And we need to be developing concrete ways of building biodiversity capacity and infrastructure for studies in the global south. As Marcelo explained, the vast majority of these collections are very underfunded. Next slide. The INSDC, the International Nucleotide Sequence Database Collaboration, recently released statements regarding the importance of open sharing of viral sequence data, specifically COVID-19, to enable research and response to the current global pandemic. But viruses and pathogens do not exist in a vacuum. Comprehensive understanding of hosts, environments, and their interactions, as is acknowledged by the One Health framework, is possible through museums. And MIPA is trying to build those connections by plugging museums into local One Health sampling networks and workflows to facilitate standard data collection, gold standard specimen or sample preservation, and broad access. Next slide. So the question is, how do biorepositories like this fit into Nagoya? Next slide. I'd argue that biorepositories fall uniquely into this gray area of being both users and providers. In fact, they're more like facilitators. Next slide. Providers can include researchers, agency biologists, veterinarians, epidemiologists, literally anyone sampling wildlife from countries all over the world. And all of these individuals can archive legally collected samples with biorepositories. Next slide. This benefits the provider by offering gold standard long-term sample preservation, access to taxonomic expertise, which is super important when trying to identify the zoonotic reservoirs of a disease, and tracking the usage of these materials. When depositing samples of the biorepository, there's typically a formal transfer of ownership from provider individuals to the biorepository institution. Next slide. And this process typically includes signed documents that indicate prior informed consent and mutually agreed on terms of use, or PIC and MAT in the language of Nagoya. Although historically, biorepositories may have used different terms. In this way, museums are already doing exactly what Nagoya mandates by facilitating mutually beneficial global access to samples for non-commercial research. Next slide. The next step is to better connect these biorepositories to other biodiversity infrastructure, including remote field stations, government agency workflows, public health officials, and local communities. The collaborative contributions from all parties that are sampling wildlife are important for building spatial, taxonomic, and temporal representation locally, and then using those resources in research and communicating the results to the public. Next slide. So we recognize that there is a long history of colonial and exploitive actions that should cause us as scientists to reflect on how to approach DSI issues moving forward and find and implement equitable and just solutions towards benefit sharing. What can we actually do? Mostly, you need to contact your national negotiators. The message can't just come from the North. It's equally, if not more important, that this message comes from the global South to reinforce the value of non-monetary benefits sharing, the importance of access to genetics and DSI data, and the distinction between commercial and non-commercial research, as well as the potential for biorepositories to meet this need. Unfortunately, there will be no more open consultations on Nagoya, which means that political decision-making will be happening behind closed doors. That is why you need to independently or as institutions approach your negotiators. And larger organized groups of international stakeholders like MIPA and other scientific societies like those sponsoring us today need to be united in these goals. Next slide. And I think it's with the support of all of today's speakers that I'd like to thank the American Institute of Biological Sciences for inviting us here today, NSF for funding this workshop, Spinach, ASM, and NSCA for sponsoring today's workshop, and Project ECHO and the MIPA Organization and Planning Committee. Because without them, MIPA wouldn't exist. So next slide. Thank you very much.
Thank you very much, Jocelyn, and all our speakers, Luciana, Marcelo, and Gerardo. At this point, we'd like to give the audience opportunities to ask any questions. So if you do have questions, please use the Q&A feature and type in your questions to our particular speaker or just generally, and we will find the best person to answer that. In the meantime, I will introduce our facilitators for the structured discussion part of our program. We have John Bates, who is part of our organizing committee. He's the Associate Curator of Birds and Head of Life Sciences, Science and Education at the Field Museum. He's also the President of the Natural Science Collections Alliance. And we have Joseph Cook, who is the Regents Professor of Biology, Curator of Mammals, and Associate Curator of Genomic Resources at the University of New Mexico's Museum of Southwestern Biology. And we'll just wait one more minute and see if there are any other questions from the audience. We do have a question from Ignacio Videla. Which are, in your opinion, the main benefits of the Nagoya Protocol? Is there any speaker that would like to take that on?
I can start. Joe looks like he has some comments too. I think the fact that this issue is at the forefront is really important. I think there needs to be benefit sharing, and the fact that we are now thinking about it in more critical ways is really important to scientific progress to make sure that other countries are not being actively exploited. So I think that's the biggest benefit. I think maybe the problem is that our lens is too narrow. We're too focused on financial benefits. And of course, governments are focused on finances. That's what they care about. But as scientists, we care about training people, educational resources, and access to data and information that can help us learn more things. And so I think there are great things about Nagoya, but maybe implementation is a little narrow.
Maybe to chip in some areas, it helped to harmonize the regulatory landscape. So you have specific approaches for access, for example, under the Sea Treaty. But for access to normal genetic resources, the Nagoya Protocol foresees some sort of harmonization, but because the interests of the providing countries are so hugely diverse, they didn't manage at the moment to conclude to some sort of common ground. What is clear now from the discussion on DSI, and I think, Jocelyn, you pretty much nailed that, this black and white painting and north-south divide simply is not there. And I think that is also a good sign. And we can immediately see that the discussion to find a solution takes so long. So the negotiators have become aware that there is no quick solution, and that they need to be very, very careful not to choke down basic research. That's the one thing. The other thing with funding and maintaining basic research infrastructures, because of the challenges that are already there, and climate change might be only one point there, there will be a lot of monitoring needed under the post-2020 global biodiversity framework. And it is pretty clear that many countries will not have the means and the capacities to fund their infrastructures. And so that is a little bit behind that discussion on money.
We have a few other questions. We had an anonymous attendee say, I like the idea of viewing biorepositories as facilitators instead of users, but what do you think of the situation where national authorities treat biorepositories as initial users with conditional MAT regulating downstream use?
So I was one that positioned biorepositories as a facilitator. I agree. I think it's a nice idea. I think we have to navigate that. That's kind of the next challenge, is how do we work the transfers of ownership here with regulators to make sure everyone's in compliance and justly sharing benefits. So I don't have a great answer. I didn't know if, Dirk has a question. I didn't know if John Bates wanted to speak up.
I think it's an interesting challenge for us, and this is why these discussions are going to continue for a long time as this goes on. But I think there are solutions out there, and one of the things that I like about the idea of MIPA and some of the ways we've discussing some of this is, if we pull off what we'd like to see happen, we would increase the capacity within Latin America for biorepositories to expand. And if we do that, then I think there's much greater capacity to see the ability to develop those mats in ways that are less probably stringent with respect to some of the restrictions.
In some countries, you can see that biorepositories take that legal obligation of serving as a national access point. Indonesia, LIPI is one example. But there's also the approach in the European Union that especially microbiological collections can register as a collection, facilitating access. I posted the information in the chat. The other option is what a lot of natural history museums in the EU did, or in Europe, so it's not only the EU. The EU offers the opportunity that you recognize best practices. So policies, procedures, and workflows you develop to support not only your own researchers, but also visiting researchers. And especially if we are talking of pathogens, we have to keep in mind the specimens that we have in the collections have been inventoried as birds, as mice, as whatever. And usually the viruses are associated with the specimen, and normally, or in many old cases, the paperwork is not sufficient and does not cover the associated organisms. So that is something new that we need to look into. And on the European side, there is a group of natural history collections, CTAF, and they developed a code of conduct that in principle guides institutions. There is a best practice part, and it's basically like a tick list where you can just go through your policies and just check if you need to recalibrate or rethink your workflows to address your compliance questions that you might have.
I think we have time for one more question before our facilitated discussion. DSI published in GenBank is in some ways obscures the difference between non-commercial and commercial use, as anyone can freely access this information. How do we simultaneously advocate for DSI not to be included in Nagoya so as not to stifle research, but also solve this fundamental problem? I guess the hardest question for the end.
Well,
Go ahead, please.
No, go ahead, Luciana, please.
Yeah, that's the key question. I don't think anyone has the answer. If you think of genetic resources as only a material good, then usually what you do is when you give permission to access genetic resources for non-scientific purposes, you give the permission and then you also insert a clause that says that if that genetic resource is going to be used for commercial purposes, then you have to come back and ask for a new permission. In that case, it's also in question, what would you do when you transfer, if you transfer that genetic resource to someone else, if those obligations would change, continue, you know? But it's harder if you think of genetic resources just as a material resource. But if you think as an informational resource too, it's very tricky because it goes to the question of information itself, music, movies, whatever that is intangible. How do you keep it close? How you keep it in a way that you can exclude others from use? So as you cannot change that fact, but also you have to hear claims of justice from different countries, there are different policy options that are being under debate, and that would have to do with triggering Article 10 under Nagoya that talks about resources that are cannot be linked to a specific country, or resources that are in areas beyond national jurisdiction. Information is a resource that could be considered beyond national jurisdictions. Once it's there, once it's stored, it's very difficult to exclude others from its use. So you have to think how you will want to share common benefits, because at the end of the day, if you think that ABS will help biodiversity conservation, then you want to make that link. A different question that I have not seen any paper answering it is the question of whether ABS is helpful at all in economics terms for biodiversity conservation. But that's a different question. But perhaps we would not be debating this if we would answer that question, if it helps at all to the objective it was meant to help. That's a different question. Thanks.
I would like to add, I think we've approached some of these issues regarding intellectual property. So I know there's been discussion about how to make claims on intellectual property and how that moves across borders, and that's the same idea as information sharing and DSI. So maybe there's an opportunity to draw from what has or has not worked in the way that we deal with intellectual property.
Thank you.
Note, you can add a data use statement. So if DSI is linked to accessed genetic resources, so that informs third on the condition under which the original material was accessed. That is one option. But the majority of DSI data sets and also the genomic information that is encoded in there is shared widely among organisms. And that's the tricky part in the DSI discussion. So the data sets that are available on INSDC, and especially if the definition of DSI presumably will get wider and not too narrow, then you simply have the problem that you cannot locate the geographic origin. And that is a legal challenge. That's the one thing there. And the other is directly linked, it's the temporal scope. So how long do you want to go back? So when was that genetic trait acquired? Can you put that into a perspective with the CBD or Nagoya Protocol? Surely not. And that raises fundamental questions for the functioning of the CBD and the Nagoya Protocol as such, and we need both to facilitate access to genetic resources, and that is really a tricky part. You have to resolve that without choking off the other.
Thank you very much. At this point, we're going to transition to our structured discussion, and I'm going to pass it over to John Bates and Joe Cook, who will lead us.
Thanks, Josie. I guess I'll ask the first question to the panel. I mean, one of the things we've been tiptoeing around here a little bit is what are the issues with respect to non-compliance for folks, and this is something that comes up very regularly in the United States because we aren't a signatory to the CBD, and so I'd like to put that out to the group and get people's responses. And Luciana, do you want to take that on for a second?
Sure, John. Yeah, being a signatory means that the United States is not a party to the convention. It's only a country when it ratifies the agreement. But still, as Josie said, researchers are doing research everywhere. So one thing is international law, and the only thing that would, let's say, the only difference would be whenever a researcher goes to a different country, of course, he or she has to comply with every law in that country. So, for example, if Brazil or Argentina or any country says and considers that genetic resources are also have an informational component, you will have to ask for permission for a PIC to access that information. The thing is that when you go back to your country, if international law does not recognize DSI under Nagoya or the CBD, then you won't be obliged to implement compliance measures in regards to DSI. So it's the same case that happens with what Dirk was saying about the recognized collections in the European Union. Each country or each bloc has the right to interpret international law the way they do. So for some countries, genetic resources are the tangible and the intangible component. For the countries that is not, they won't apply compliance measures for this. The problem is not the law. The problem is that the issue complicates life for researchers. That's the issue you want to avoid.
Anyone else want to?
I can say what's here in Brazil, what we are bound by the law as Brazilians. I know that non-Brazilians are not bound by that, but here we'll be fine if we don't follow the law. So friends here in our institutions are fined in thousands of reais, and the institutions themselves can be fined in millions of reais if they simply don't state the correct term in their package, they can be fined, and it's hard to get those fines reversed.
There have been first cases where journals have been approached by provider countries after publication of research results, where the competent authority informed the journal that the permits that were issued to the researchers were not proper to cover the research that has been done, which led to retraction of the articles. And I think that raises a visibility there for the researchers, but also for their institutions, which is not too healthy, especially if you have to apply for funding. So, and of course, there have been cases in the past where also some NGOs jumped in, and usually they are out for the iconic names. And so what individual users, which is scientists and researchers, need to understand, I think, is that beyond their own research intention and focus, they have a bigger responsibility, and that is at least for the institution and the colleagues at that institution. Because if the institution is exposed, and this is usually what happens because all the contracts obviously are signed by the institution, not by the individual researchers, simply put the institution in a very bad position if that spreads to negotiate new contracts.
Okay, great. Let's move on to this next question. Actually, I'm going to jump to the third one here. How do you connect organismal collections in DSI? I guess that's a question maybe for the biologists there on the call. Or do you?
Yeah, my answer can be biased. I think the voucher, the specimens, is the central part of the connection there. They should have how do you say a permanent indicator, a PID, and then relate that when you deposit sequence in gene bank. I mean, we've been doing that for a while. And we've been recommending with the NCBI to make that mandatory. Maybe that's the time to try to put some pressure there again. I don't know if that was the answer, the direction, Joe.
I think so.
And Marcelo, I just add to that you know, this particular example that we're talking about here with disease-related research is something where we still don't know, for instance, where in the environment COVID specifically came from yet. And there are some real questions, as I know the mammalogists on this call know well, associated with what the taxonomy is of the groups that are involved in potentially the transfer. And so I think this is one of the things we keep saying, obviously, is that for the COVID pandemic is a fascinating lesson in the value of potential voucher specimens. And Gerardo, I don't know, do you want to say some more about that with respect to your research?
Well, this is a great opportunity to rethinking our relation with nature, no? This is a great opportunity to rethinking how we procedure in development, but also how we deal with natural resources and genetic resources. So this is a very unique opportunity, no, to enhance collaborations, and this like MEPA and all these webs are very, very nice ideas to continue in a good way to react, because if we work and we continue as usual in all aspects, the same happen. We'll have again. We'll have pandemics. We'll have other problems. The same habitat destruction, biodiversity loss. So this is a good way to enhance all this collaboration in this topic, in genetic resources. Basically, I don't know, is what I think.
With regard to collections, I think, Marcelo, you did some research on the number of viral publications that were actually tied to actual collections, right? And what, you remember what that was?
Yeah, we did a review on the papers that were dealing with coronavirus and their reservoirs, and a single paper out of how many were there? About 50. Only one had proper voucher information, proper vouchering, actually. Several papers, they just don't mention vouchering at all, or do not collect a few specimens as vouchers for biorepositories. And that we actually, I was going to put the link. We had a paper mentioning that, the one with Cody. And I think that's something not easy to correct, but something that we should always keep in mind when dealing with collecting. And then the connection, the permanent link between a voucher and the DSI. That was the beginning of the question. We need to establish that link in the institutions.
Isn't that a general concern that molecular results are only published by GenBank numbers? And with regard to viruses, for example, if you lose the connection to the organism and all the metadata that is available for that specimen, isn't that a bad idea? I think that is something that fast science need to rethink there. Also, speaking of good research, your results should be testable by others. And for that, you would need that information. And the smarter you are moving there in that landscape, I think the more difficult it gets to really come back to the original specimens. And I think good science just needs that. And we don't need to touch Nagoya there. That is a key principle in research.
I want to jump in here because I've had the opportunity to see some of these working groups, international working groups that are trying to connect these open databases so that you could go into, for example, the Global Biodiversity Information Facility, see where specimens are. From the specimens, you see where there's genetic sequence data. And we have so many tremendous databases that we need to link up. And there's a lot of really great people trying to put in this effort, and they're doing it tirelessly and often without enough funding. And it's really amazing. And if these linkages can be improved, and I have hope for these people to be able to make some headway by coordination, that a lot of this will not be the burden of the researcher. But we also do need enforcement. A lot of journals still don't require that you mention the vouchers or that you show that you have permits. And so that's kind of also a problem, is there's really no rules right now.
A-Start Molecular Ecology just did that. So it's coming down the road. And if we see the Nagoya Protocol positive there, Nagoya simply tells us, get better.
So the discussion of databases is good. And in the chat right now, we have an interesting discussion that points to a lack of benefit sharing with regard to what's just happened with SARS-CoV-2 and publishing sequences. So Enrique Lessa is pointing out that some of those folks that published their data to these databases then were scooped by larger research operations that were able to do that. So we're going to talk about benefit sharing here in a second. John, did you have another question there to lead us on?
Unmute myself here. So how has Nagoya changed the way you're doing research? And how do you define responsible use and implement it?
I think this is a great question because it really highlights A, how different regulations are in every country, and B, the gray area surrounding responsible use. As a curator, we weigh responsible use with every proposal that is submitted to use our collections, recognizing that these resources are limited, especially tissues. We typically have a small portion of a tissue in a tube, and we want to make sure that we get maximum amount of information and that the researchers are qualified and that it's going towards non-commercial research. So I guess there is a lot of gray area there. For how it's changed my research, I can't say tremendously, but it makes me glad that I do have research in my home country because it's easier. That's bad to say, but it's true. Gerardo, how has it changed things for you?
Oh, it's a challenge because, for example, in Mexico, I guess in general, the viruses and bacteria and prions should be included as a biodiversity in the biodiversity collections. We have a challenge in Mexico to include all the information, genetic diversity of viruses and bacteria, to include it as a biodiversity, and to work together with biologists and the health people to include this. So I guess this is a challenge to include this, even for the health department, animal health department, to include this and view it as a biodiversity. This is one of the challenge. But this Nagoya Protocol, well, is changing in different ways the way we are working on collecting data, sampling animals has been changing in the last 3 years. Now we are thinking in a metagenomic way. We are thinking in another way of collecting samples, and it's a new dimension of biodiversity. So it's changing in different ways, and this offers an opportunity to work in another way, international collaborations. No? So I like all this input we are right now.
I think, I want to bring up one of the comments in the chat. Liliana Davalos points out that unrestricted public access benefits those who already have access to technology. And I'd like to hear the group's thoughts on that.
I would say that what Liliana is saying is actually the claim that provider countries do, because actually, in general, in the discourses of user countries, of course, all countries can be users and providers, but in reality, there are countries that are more of a provider and more of a user, right? All things are real. So let's just talk about them like that to be clear. When in the statements that user countries do, the benefit that they always say they will have is that sharing information is good for everyone. Sharing open access is good for everyone. And of course, that would be good in a world where everything is shared, but that is not the world we live in. So this is actually, with an easy example, like giving a book to someone that cannot read. In this case, lifting tides do not lift all boats, right? So you need to create capacities. You need to transfer technology to be in order to get equity, and then you can say, okay, everything will be shared. That is just the question. I mean, it happens in every country. In every country, you also have people who have more and who has less. And the people who has less, perhaps, were not able to get vaccinated, even in poorer countries, as people that were rich. So it's always a question of how you can make the less advantaged be a little bit better in order to at least get a little bit of a benefit out of the entire thing. And also how to really allocate these benefits for biodiversity conservation. Otherwise, it doesn't make any point to be arguing about this if we lose the focus of the issue, that is biodiversity conservation. It was thought to be for that, ABS.
To add a positive comment there on the Nagoya Protocol, I think it is very useful because it clearly shows us the gaps. And the gaps there are not directly linked to the Nagoya Protocol because most research today is based on funded projects. And researchers in developing countries have the same issues to get the paperwork timely than those in user countries. And as we also talking about linking information and linking information back, I think what the Nagoya Protocol actually does is it shows us the existing weaknesses. And those weaknesses have been there before, but we haven't looked at them. I just mentioned good research. So that we have the problem that our GenBank numbers are not linked with our database entries is nothing new. That existed before Nagoya. So I think, I know we all complain quite a bit on the Nagoya Protocol, but if we try to be a little bit more positive there, I think we have a great opportunity there to improve what is needed. And if we want to look into pathogens, we need the metadata. Without the metadata, the information is pointless.
So one thing we want to spend some time on in this discussion is brainstorming about benefit sharing and what that might look like. And I think we've already hit on some of that here in terms of what can the scientific community offer in terms of benefit sharing to providing countries. And what would that look like? Because I think if we're asking folks to talk to their negotiators, that they need to be able to propose some solutions, some actual things that could be included in language, right? And so I guess I'd throw this out to the group. What are some of the best practices you employed during your collaboration that you think others within the scientific community should replicate with respect to sharing the benefits of your research? And we've got a couple of other questions here too. Let's start with that one.
One practice? Oh, sorry.
Go ahead. I think that the first one is tell that you are sharing benefits. It's so simple. We came up with a list. So we were asking our researchers as part of the annual report simply to name their collaborations and what they have been doing. You can see that list on our homepage. It is impressive. We never thought that, because on a daily basis, you don't talk to your colleagues, but it was impressive what came up out of all these research collaborations, and nobody knows. And again, the good thing here is for the post-2020 global biodiversity framework, there will be monitoring elements for benefits that are shared, and that is both commercial and non-commercial. So again, huge opportunity to increase the visibility of non-monetary benefits that are already delivered. they are not visible at the moment, and that is a huge issue.
I have an add-on question. What about across borders when there's one mammalian species, for example, that is across two borders? Are there benefits just by having countries talk to each other more about conservation or regulation?
That is even a more complicated question, because the law says that in that case, countries should cooperate, and most of the species are non-endemic, right? They should cooperate, but this is not the case. So what usually happens is that researchers do green shopping, right? They go to the country that has laws that are clearer or easier or do not have any law. So this is also something important for authorities to understand, that it's not like they have resources that are just in their country, but they are spread among different countries usually, and that if they get too restrictive with their own laws, they won't get any benefit at all. So they need to keep a balance on making legislation.
And I was just going to say that with respect to access and benefit sharing over the course of my career, one of the things I've come to value the most is an obvious one, which is training. But when I say that, one of the things that I've loved seeing is cross-country training in the global South and how much that's grown over the time that I've been doing science. And we've done whatever we could to try to find money to do that. But I think that in a sense, what Rachel, when you described the small mammal, I think it's just as important for humans to realize that there are huge benefits from understanding what the country next door to you is doing and how they're doing it. And that's been just like a mantra in the back of my mind the entire time I've been trying to study avian biodiversity.
I would say that it ties into the concepts that Jocelyn brought up in terms of sort of scientific colonialism, right? And so for me, if I'm going to go and work in a place, I'm going to dig in and get to know the people and the institutions and have equal, scientific partnerships. And it's so much about infrastructure development. It's me constantly asking at a group meeting this morning, what can I do for you? Do you have any questions for me? What can I do for you? When we're there, can we run some trainings? Can we bring your students? It's just, I think it's the benefits to using those data sort of just fit part and parcel with a shift in the way that we are working in general in the global South, or at least that we should be.
I would add that another way to do benefit sharing, non-monetary benefit sharing, could be to repatriate the information that has been obtained. Sometimes when researchers leave the country, they don't get back that information. And even more important, that information makes sense and is relevant for the country of origin. That you can go to make it easier, for instance, for conservation, right? Especially if you are in a project where indigenous or local peoples are engaged, to repatriate this information in a way that is culturally appropriate for them and contributes to conservation is very important. And as Diane said, it's also important to ask the local people, what do they need? Perhaps it's not what one thinks they need, but when you ask them, it's the clever thing to do. Just to make a quick example, I was involved in a development program and a development project in a country from Africa. I won't mention the name of the country. But that country has gone 20 years of civil war. And at the moment that we were doing the project, they only had 7 students who had a PhD. Only 7 people in the country had a PhD. So our project was about ABS and capacity buildings on law and these things. So we had a budget for buying books. Of course, we bought some books on ABS, but then we asked the students and their teachers, what books do you need? And they asked for books that were, I'm sure, I'm not a biologist, but I'm sure that they were books that are standard, basic books for any biology student in the North, in Europe or in the United States or Japan. So if we would think that we knew the answer, we would have failed. We had to ask them, what do you need? We got the answer. Sure that if you go to different countries, they would say, we need softwares, we need management systems, we need training, things that maybe, of course, they have a cost, but it's not money, it's this contact. Even joint publications for researchers are benefits.
I'm going to jump in one more time, and that's because I'm inspired from this conversation to wondering, can we as researchers also have some benefit sharing by communicating back to those focal points, the Nagoya focal points in other countries, and relaying the experiences or the people that really, through our own collaborations, we've discovered should receive benefits, so that we can help train and prepare the Nagoya focal points to be better equipped to work with the next people. I've written many Nagoya focal points, and usually I don't get a response just with simple questions, because there's just so much complexity and so much still in development. I'm not sure if you all have experiences working with different focal points, but I wanted to bring that up.
I think the question there is, so the obligation to share this information, that's nothing new either. So that's in place, and for some countries that established access legislation under the CBD back in 1993. I think the key problem there is that individual scientists should understand the contracts they sign. And in principle, we touch the same issue there, because if you loan material, a standard phrase in many loan agreement forms is that you should share publications. And I don't think I need to mention the war here. That is a long history. And again, the Nagoya Protocol shows us an obvious gap there. And I think there is huge potential with the digital opportunities that we have. If you look at GBIF, for example, so taking that up and making that available in a public domain and improving the country tags so that the information can be perceived from the providing countries is something good. On the other hand, and this is what we can learn from the tracing and tracking that is already in place, that provider country does to identify cases of non-compliance, it shows us that this is working the other way around as well. So if you don't meet your contractual obligations, there might be some issues coming down the road. So all should be aware, if they publish, they have a visibility, and that is not only the key audience there. So it should be in our own interest to improve that. The point there is, if you talk to DSI, we are talking here the entire time of INSDC. Yeah, so that is the data that is in the public domain, but that is not that DSI where the commercial revenue is generated, because that usually hits in closed databases. And the huge issue there is everything, all the policy options that are discussed at the moment are tailored or discussed around INSDC. But there is, at least maybe Luciana can help me there, but I don't know any international law that would oblige commercial users to disclose the information in their databases. So that is an issue. And so you have to be aware on the political debate, so that there are a lot of smoke screens there. And of course, it is a concern with INSDC data there. But that might not be the key question.
I'd like to just ask one last question, because we've got this session sponsored by societies. And that is this question of what is the role of scientific societies in fostering ABS? I think that, societies are where scientists organize, and we haven't often taken on issues like this as societies. What is our role? Should we be more active in this sort of thing? Any thoughts out there? Okay, we already know about you, Dirk. Anybody from the Mammal Society want to speak to that?
I recognized there were a whole bunch of names of folks on the call today that I recognized, and some other life members of ASM. And I feel like it's maybe the first place that people turn. So is this as a member of this organization, we talk all the time about what are the benefits of being a part of a member organization. And one of those things can be, I think, really helping people navigate these issues and having resources and places to point people. And I think that for many societies, especially I think the older ones who've been around for a long time, there's some real pivoting that still needs to happen to facilitate that.
I would ask though, Deanne, if it's even something more than that in terms of informing our members, but maybe having the society actually weigh in with our negotiators, or having the society organize in a way that's more proactive.
I agree, Joe, and especially because the societies can bridge the national societies, they have relationships among them. So this have to be confounded, they have to be integrating among the different countries. So it could be very important, yes, to have those transnational collaborations through the societies.
I agree. I think one of the things, the inertia components, is that they're largely everyone, we're all scientific volunteers in this way, right? And so more like it's, well, maybe we can provide the funding to, fund a half position that we're there sharing with other societies so that we can move this forward in a really proactive way.
Okay, we're at the magic moment.
All right. Well, thank you, everybody, for that excellent and enlightening discussion. I'm certainly encouraged by all the ideas and suggestions that were shared today. As we approach the end of our program, I just want to reiterate that this is only the beginning and not the end of our conversation. And we can continue the discussion and share ideas on the workshop discussion portal on learnnagoya.com. This will also be where we will be posting today's recording and other resources and materials that were shared over chat. And please feel free to share those with me or put them in the chat, and I will make sure that they are collected and posted on the Learn Nagoya portal. And please take advantage of this community space and share your ideas and perspectives. We want to hear from you. And I'd also like to share that we will have several more workshops coming up in this series, as you can see on the slide. So if you're interested, please do sign up. Note that the next one is this Friday. So please visit our event page, which is io.aibs.org/Nagoya to learn more and to register for any of these sessions. We hope to see you again in future workshops. And just in closing, I want to thank all of our speakers and workshop facilitators for their time and input and for sharing their perspectives today. And thanks once again to our host societies, ASM, NSCA, and Spinach for supporting this event. And none of this would be possible, of course, without funding from the National Science Foundation. So thank you, NSF. And last but not the least, a big thank you to all of our attendees today for joining us and for participating and for contributing through chat. We hope that you will continue to engage in these discussions with us, with each other, with your colleagues, and your policymakers and focal points on these issues. Thank you, everybody, and have a great rest of the day or evening.